Good. So hi, my name is Avital, and I'll be talking about how the blind see. Spontaneous brain activity underlies spontaneous perceptual behavior. So spontaneous brain activity is basically what happens in our brains when we do nothing. And we know quite a bit about it. We know it's ultra slow. We know it appears across the entire brain, but we don't yet know what its functional role is. One hypothesis brought forward was that spontaneous brain activity may underlie conscious cognitive behavior. So we usually think about our behavior as being evoked from some outside stimulus. For example, if you look at this purple pink thing here, this could be our stimulus. If we encounter it, then we would have some brain activity. And if it's sufficient, then it would cross some theoretical activation threshold. And then we would enter into an active mode. That is, we would show stimulus-related behavior. But it has been proposed that these very slow spontaneous fluctuations could occasionally cross this activation threshold. And by doing so, we would behave, but not because of some outside stimulus, but because of internal stimulation. This is what I will call unprompted behavior or spontaneous behavior. Now, it's very hard to really test whether spontaneous neural activity could underlie unprompted behavior for two reasons. The first is that you simply cannot ask participants to behave spontaneously, right? It loses all spontaneity. On the other hand, if you're looking at brain activity, then once participants behave, then their brain activity is not spontaneous anymore. It's task evoked. So to overcome these problems, we decided to study late blind individuals, because there is no external vision evoked activity in their deprived visual cortex. So their visual cortex is activated spontaneously. But we chose those individuals that show very vivid visual hallucinations, as vivid as they used to see before. And because neither the appearance nor the content of these hallucinations is controlled by the participants, these are genuinely unprompted perceptual behaviors. Now, this condition is termed the Charles-Bonnet Syndrome, CBS, and is quite rare. But we managed to recruit 5 participants for our study. The first one hallucinates a face, a male face, that appears and disappears across her visual field. The second one sees vague faces that he describes as ghost-like or different patterns. And the third one sees still images of various objects and people. Now, these 3 participants can very accurately describe their hallucinations, both in the temporal and in the spatial domain. But we also recruited 2 more participants who couldn't give such an accurate report because their hallucinations actually spanned the entire visual field simultaneously and very rapidly. So one saw flashes of light and the other one saw patterns that look like a kaleidoscope. We also recruited 11 late onset blind controls who have never hallucinated and 13 sighted controls. Now, what are the neural correlates of hallucinations in CBS? This has been previously studied, and previous work, for example, Fitch et al., have actually ascribed hallucinations in CBS to very focal activity in the visual system in an area near the fusiform gyrus. But if we think about the visual system, we know it's highly interconnected. And we know that at least when we see, then any activity that is sufficient to evoke vision doesn't really remain focal in one ROI in the brain, in one area in the brain. It spreads through the visual system. So unlike previous accounts of CBS, we first hypothesized that visual hallucinations, like veridical vision, would activate the entire visual hierarchy. Now, another controversy in the literature is that some studies have actually proposed that hallucinations in CBS are evoked from externally evoked stimulus. So they are evoked by the external environment. But this Fitch et al. Paper actually showed a slow buildup of activity before the onset of hallucination, which makes the possibility of this being triggered by the outside a bit tricky. So this actually aligns with our own hypothesis that spontaneous activity accumulates and brings about hallucinations. So we have another temporal hypothesis that hallucinations would differ from veridical vision in the spontaneous buildup of activity prior to perceptual events. Now, importantly, we actually hypothesized that this would happen across the entire visual system, not only in one part of the visual system. Now, to do this, we asked our CBS participants to give a verbal report of their hallucinations while their brains were being scanned in the MRI scanner. We know exactly when hallucinations occur. And we also recorded these reports because we played them back to the participants after being scanned. And we asked them to give as many details as they can about the content of these hallucinations. We then used these logs and descriptions to create simulated hallucinations, movies that represented decided controls in the MRI scanner. And we asked these controls to also describe what they see. Now, I will give you some example of the hallucinatory streams that we created. This is the hallucinatory stream of the first participant who hallucinates a face. This one is of the second participant who sees patterns and ghost-like faces. And this belongs to the third participant who sees still images of various objects. Now, we have two experimental conditions. We have hallucinations, which are spontaneous vision, and we have veridical vision, where the vision is cued by stimuli. But these two conditions actually differ in another way, and that is that hallucinations are internally generated while veridical vision is externally generated. So to further isolate the spontaneous component of hallucinations, we introduced another condition, visual imagery. Because here the vision is cued. We tell participants when to visually imagine things, but the vision is internally generated. So again, across all these types of vision, we expect to find a very similar pattern of activity spanning the entire visual cortex. However, in the temporal domain, we expect that for the cued types of vision, first the stimulus or instruction would appear or be given, and only then the signal would rise. Whereas in the spontaneous type of vision in hallucinations, first the signal would rise and only then the hallucination would appear. So what do we see in the brain of CBS participants? Well, on the right, you can see an inflated brain, and on the left, just the flattened cortex. And you can see that these maps are not thresholded because I wanted to you to appreciate where we actually find activity. In red contours, you can see areas that are statistically significant and activated during hallucinations. They're also corrected for multiple comparisons. And you can see they nicely span the entire visual cortex. So hallucinations actually activate the entire visual cortex. Now, what happens during veridical vision in the simulated hallucinations? Well, we find more confined activation to the fovea. And this is probably because our stimulus had to be small enough to allow movement on the display screen. So we didn't really activate the periphery of the visual system. Nevertheless, if we superimpose these red contours of significant areas of hallucinations onto the controls activation map, we find some level of overlap. And if we actually correlate the patterns of activity between individual subjects, CBS or sighted controls when they see, we actually find a very significant level of overlap. So it seems that hallucination and veridical vision share some spatial components of activation. Now, this is again the hallucination map of CBS participants. And now let's compare it to visual imagery. This is visual imagery in the CBS participants. And again, you can see that we have a lot of activation in the visual system. And if we superimpose the hallucination contours on the imagery map, we find a very nice overlap. But this is a very small sample, so we can do a very good statistic on it. So because CBS comes from the blind population, then we can just look at the imagery map of the blind controls. And here again, you see a very similar map. And again, if we superimpose the hallucination contours onto this map, we find a very nice overlap that is also statistically significant. So it seems that hallucinations, like other types of vision, activate the entire visual hierarchy. Now, what about the temporal hypothesis? We thought that spontaneous activity would accumulate until hallucinations would appear. To test this, we chose 3 ROIs: early visual cortex, V1 to V4, the FFA, and the lip area. And the lip area is a control area because it is engaged in the task because of the report, but it's not part of the visual system. Now, here you see the onset of hallucination or stimulus. And the CBS participant in green, you can see that their signals actually rise before the hallucination appears, which is not the case for sighted controls in black, where first the stimulus appears and only then the signal rises. Now, importantly, this is unique to the hallucination condition because there is no group difference during imagery. We see a very similar effect for the FFA, although reduced in its amplitude. And importantly, there is no effect, no group difference for the lip area. So this effect is not only specific to hallucination in CBS, it's specific to the visual system of individuals with CBS. So hallucinations differ from other types of vision in the buildup of spontaneous activity prior to perceptual events. Now, as you can see, there is a difference in the level of effect between early visual cortex and FFA. So we wondered if this signal accumulation is uniform across the visual system. To understand this, we ranked visual areas from an atlas based on their position in the visual hierarchy. And we then correlated these ranks of ROIs with the time in which the signal rises relative to the onset of hallucinations in CBS participants. So the more negative you see a dot here, it means that the more preceding signal there is before hallucination. And as you can see, there is a very nice and significant correlation showing that the accumulation of signal is most pronounced in early visual cortex, and then it decays along the visual hierarchy. Now, you may wonder how we could measure such a thing using, given the sluggish nature of the bold signal. This is because, as I said before, spontaneous activity is ultra slow. So unlike visual evoked activity, we can actually see this using the bold signal. Importantly, this effect does not exist in the imagery condition in the CBS participant. So it's not some trait of the CBS brain. It is simply related to hallucinations. So anticipatory spontaneous activity decays along the visual hierarchy during hallucinations. Now let's sum up. Here we compared 3 types of vision. We had hallucinations in CBS, simulated hallucinations, veridical vision inside of controls, and we had visual imagery in the blind. And we've shown that in the spatial domain, visual hallucinations, like other visual experiences, engage the entire visual system. However, in the temporal domain, only in the hallucination condition, there is a buildup of bold activity prior to hallucination onset. What we believe is happening here is that in the deprived visual cortex of CBS participants, and specifically in their early visual cortex, some spontaneous activity may occasionally cross some activation threshold. By doing so, it would activate the entire visual system by propagating through the entire hierarchy. And this, just like typical vision, would bring about the experience of vision. So in other words, spontaneous brain activity can evoke vision or perceptual behavior. And if so, it's tempting to think that perhaps spontaneous activity may drive other types of unprompted behavior. For example, when we close our eyes and go to sleep, our visual system is deprived, and therefore any spontaneous fluctuation in early visual cortex may propagate and ignite our visual system, thus bringing about the experience of visual dreams. I would like to thank my collaborators on this work, Meital Wilf, Boris Rosin, Marlene Berman, and Rafael Malach, the different funding agencies that support my work, and I want to thank you for listening.
Thank you very much, Avital. So, hi everyone, I'm your backup host, Emcee, for today. Nice to meet you all, and thank you very much for your talk. So we have many questions. I think we can take some online. So the first one is from Mohammed. What is the unit measure of signal accumulation? What exactly is accumulating in a given region?
So when I say accumulation, what I basically mean is the rise of the bold signal. So what we were measuring here is if a hallucination appears in a certain point in time, whether we would see that the bold signal would rise prior to that hallucinatory event. So by accumulation, I basically mean the rise of the signal until its peak.
Perfect, thank you. Then we have another question from Haida. Did you see any differences between the people who experienced face hallucination versus lower level visual hallucinations, for example, in the FFA?
Yeah, so interestingly, across our CBS participants who had very different visual experiences, we still saw the entire visual system lighting up. We couldn't find any specificity of hallucination of activity to the content. So it's not as if faces, hallucinating faces activates the FFA more. That wasn't the case. So that seems to be something that is more general about hallucinations and CBS rather than stimulus or hallucination specific.
Thank you very much. Then we have a last question from Ariel. How similar, different are the reports of the Bonnet Syndrome patients to other non-veridical hallucinations like dreams? Do you see the same neural activity patterns that you would for dreams?
So as far as I know, no one has actually managed to study dreams in the fMRI scanner. I can say that in comparison to other types of hallucinations, we have hallucinations in Parkinson's and in Alzheimer's disease. There are similarities and there are differences, but I think what we're looking at here is quite different because hallucinations in other conditions are evoked by some whole brain pathology, whereas here it's evoked by deprivation. So the visual system in these individuals is deprived and hence could act spontaneously. And I wouldn't expect to find a similar result or a similar type of activation in a sighted brain. As for dreams, that's a fascinating question. I'm afraid I don't know the answer for it.
Thank you very much. We have two more questions, but unfortunately we are run out of time. So